Thank you so much, Dr. Soma, for the insightful presentation. We'd like to continue the panel discussion around the testing and validation. I'd like to invite Dr. Lalita R. Gowda to the stage, retired Chief Scientist, CSIR CFTR Mysore. The session will be moderated by Dr. Lalita R. Gowda, formerly served as the Chief Scientist at CSIR CFTR Mysore, a member of FSSI, Member Scientific Committee of FSSAI. She's currently the chairman of the scientific panel of genetically modified foods. Dr. Lalita comes with 36 years of professional experience as a research scientist in the domain. She needs no introduction. Most of us present here know her immense contribution in the years she has put in the food industry. We welcome you, ma'am. And joining her are the panel members, Dr. Soma Dasgupta, GCMS Application Engineer, Agilent Technologies. Dr. Kaushik Banerjee, Director, ICAR of National Research Center for Grapes, Pune. He develops methods to detect pesticide residues and mycotoxins in food and assesses risks for crop scientific MRLs. As a member of FSSAI scientific panels, he aids in shaping India's food safety standards and chairs the India section of AOAC International. Next, I'd like to invite Mr. Rangu Rao, CEO, Safe Harvest Private Limited, Bangalore. Round of applause. Previously, Rangu sir was the co-founder at Samaj Pragati Sahayog and also held positions at the University of Delhi. Next, I'd like to invite to the stage Mr. Vijayananda BN, Research Technologist, ITC Life Sciences and Technology Center. He has been working as the Senior Research Associate, heading the Food Safety Analysis in ITC R&D Center, ITC Limited, for more than 18 years. Prior to this, he has worked for 2 years as an executive in the Analytical Department of Strides Technology and Research, STAR. Before that, he had 1 year in R&D development of Natural Remedies Private Limited as research assistant. Ladies and gentlemen, please welcome our panelists with a round of applause. Over to you, Dr. Lalita ma'am. Yeah.
Good afternoon. Good afternoon, everyone, and welcome to this session. I would like to welcome our 4 esteemed panelists for this session. We all know, and we all have been hearing since morning about food safety culture, etc. If you really look at the food safety part of it, the very heart or basis of food safety is testing, because the safety of food is based on the testing that we do, whether it is the quality testing, the testing that we do for quality, or the testing that we do for safety, which includes heavy metals, pesticide residues, naturally occurring toxins, mycotoxins, PFAS, which has now come in. And testing has become, in the past month or so, we have been hearing so much about, especially as Dr. Soma mentioned about ETO. And if you look at the controversies that have arisen because of the ETO that was found in the spice residues that were there, it all boils down to, do we really have a harmonized method for testing? Do all the countries use the same method for testing to give the result? And this is very, very important when you look at the levels at which we do the testing. The other question raised about ETO, which recently came up, is the regulatory levels. If you look at the regulations in India, ETO is not allowed at all. ETO is generally used to sterilize medical equipment. And it so happens that our spice makers are using ETO to sterilize spices so that salmonella doesn't grow, because you know that in the spices, one of the major microbiological hazards is salmonella, and ETO is being used. Coming to the fact about the regulations and thing, if you look at it, Hong Kong said 1 ppm. Singapore has a limit of 50 ppm. The European Union has a limit of 0.1 ppm. If you look at USA, they say 7 ppm for ETO and 960 ppm for the byproduct 2-chloroethanol. So there is so much of difference between the regulatory standards across the globe. And many food industries and FPOs do not understand what this is. If you look at the FSSAI regulations for pesticide residues, and if you look at the food additives, we have what are known as the maximum residue limits. In terms of food additives, it is based on safety. And the safety testing is done by JECFA, that is the Joint Committee of WHO and FAO. So it is based on a human safety, based on safety testing, toxicity testing. But when you look at pesticide residues, you look at MRLs, we do not understand what these MRLs, and we assume that it is FDO. So with this thing, I would just like to ask Dr. Kaushik to enlighten us on how MRLs for pesticide residues are set in our country or across the globe, including, everyone talks about SANTE guidelines and EU, but I think we should concentrate on FSSAI standards because all the food is for India. Kaushik, if you could just, throw some light on that.
Okay, ma'am. Good afternoon, everybody. Thank you for this invitation to participate in today's event. It's a very pertinent question. We know that with the recent problem of ethylene oxide, everyone is talking about maximum residual limit, how MRLs are set, why MRL is not there for ethylene oxide in our country, and all such sort of things became common man's question. So just to give you an idea that MRL is the maximum residual limit, which is basically allowed limit. It's an allowed concentration in or on food item. That means if a sample of any food item is tested by a laboratory, and if the residue of a pesticide is detected, and then what you have to do, you have to compare the pesticide concentration against the MRL. And if MRL is exceeded, this means this is non-compliant. So as per our Food Safety Standards Act, it cannot be sold or it cannot be consumed by the consumers. So this situation, we know that almost we have 292 or 293 pesticides which are registered in our country. And I think since last more than a decade in our country, every pesticide got registered with fixation of MRL. So pesticide is registered with Central Insecticide Board and Registration Committee, Ministry of Agriculture. And MRL is fixed by Food Safety Standards Authority of India, or FSSAI. So FSSAI has a separate scientific panel for fixation of MRL, and they work in tandem with another scientific panel that is on method of analysis. So this is very similar to the Codex system, like Codex has Codex Committee on Pesticide Residues, and another is CCMAS, Codex Committee on Method of Analysis and Sampling. So this is the typical style in India also we do in the very similar way. So there are multiple stakeholders which are involved in MRL fixation. First thing is that there should be pesticide manufacturer. Then they need to generate residue data. So for generating residue data, it is usually done at state agriculture universities or ICAR institutes, or even some of the private bodies are also now approved by Ministry of Agriculture for conducting such type of residue studies. Once the residue study is done, basically what we do, we apply a pesticide at recommended dose and 1.25 times of the recommended dose. And at 0 day, 1 day, 3 day, 5 day, 7 day, 10 day, 15 day, at different intervals, samples are taken. They are brought to the laboratory. They are tested in the lab. And once all the lab test is done, they are compiled and report is submitted to CIBRC. So the company or manufacturer of the pesticide, they submit the application, and then CIBRC evaluates that and then send data to FSSAI. So FSSAI scientific panel, what they do, they evaluate data. Now, if it is a simple vegetable, like every 3-day or 5-day, there is a new flush. For example, okra, green chili, etc. Usually third day sample is taken for risk assessment. In case of fruits or long-term, long-duration crops like grapes, mango, etc., 15-day or even sometimes, if it is a moderate duration, 7-day residue is taken for risk assessment. So risk assessment means what we do, you basically take all the data and that data is entered into a calculator, MRL calculator. That is called OECD MRL calculator. This is an internationally harmonized MRL calculator. And there, that third-day residue or 7-day residue that is entered. And based on that, we get some of the statistical information like highest residue, what is the median value, then mean value, etc. So usually for a short duration crop where residue is very fast degrading, at that time mean plus 4 into standard deviation. That is a typical style by which MRL is calculated. Otherwise, it could be the highest residue or it could be the STMR. STMR means supervised trial median residue. So if you are interested, you can have a lot of information in FSSAI website also. There is a SOP which has been given by FSSAI that how MRL is calculated in our country. And if MRL is not available, then as per codex, we take another rule, that is 0.01 will be the default MRL. So if MRL is not available, any residue content has to be compared with the 0.01 milligram per kilogram. If it is below that, then it is accepted. So for example, in earlier presentation of Dr. Soma Dasgupta, you have seen that sometimes the instrument can detect at a very, very low level. So trace level. So that does not mean that that level is toxic or harmful to us. So 0.001 is basically a level which is set as a default value if risk assessment based MRL is not available. Thank you very much.
Thank you. Thank you, Kaushik. The only thing I wanted to emphasize here is we don't have toxicity data. It is theoretical data on which maximum residue limits for pesticides are. And Kaushik has very clearly explained how MRLs are set in our country. And the major thing to note is that if there is no MRL, it's 0.01. Coming back to ETO, if you see, ETO is not registered as a pesticide. It is a post-harvest fumigant. And so, as you see, we do not have for post-harvest fumigants and things. And to date, there isn't a single human study, a feeding study or an animal toxicity study to show that ETO is toxic or carcinogenic. The only studies that are available are inhalation. That is because it is said that those who work in the medical field or sterilization, it's an occupational hazard. So the toxicity or the carcinogenicity has been showed as an occupational hazard, not by consumption. And so that is why, if you look, US FDA, they have limits of 7 ppm and 960 ppm for 2-chloroethanol. So just coming back to you, Dr. Soma, with all this residue levels going down to 0.1, 0.01, PPT, etc., and the instrumentation that you just described we know that, as you move down the scale to sensitivity loading, your instrumentation cost goes up. So how do you expect, it's going to be 3 times or 4 times? Right now, I think a GCM SMS costs about 3.5 crores, if I'm not mistaken. And so how do we expect when such things are there for testing and things? Small laboratories like our state food laboratories or other, small laboratories to adapt to this technology and to things. So will Agilent have a solution? Can you make equipment less costlier with the same sensitivity?
We can definitely make less costlier instruments. We do have less costlier instruments. But then, again, the quality word is also applicable to the instruments as well. So if we do have multiple models for the same kind of equipment. So let's take an example for GC itself. We have 3 to 4 different types of GCs depending upon the area that we are going to cater it to. So if the requirement of a particular lab is not that stringent, then there is no requirement to have a very high sensitive system. And so for mostly what happens is with the testing labs, usually they tend to take 1/10 of the MRL as their limit of quantification. So they are already 10 times lower than the current market requirement. So the lesser costly versions of the instruments that we have are already good enough to meet the MRL requirements which is there as per the regulatory. But if the user wants something 10 times below than the requirement, then of course we need to invest into a costlier technology, because most of these instrumentations, mass specs, particularly in particular, the for a GCMS or an LCMS, the mass spec is the one which has the higher price compared to the separation technique. And it is mainly because of the precision machined parts which go into it. In fact, to come up with a completely new source or a completely new design of the quadrupoles or the detector, it takes years of R&D. And this R&D involves testing across, not only across years, but across multiple locations and on multiple matrices, multiple compounds. And it also needs to pass through a certain level of CE certification and other certifications as well. So just like the food that you test needs a certification, our instruments also need to go for a certification, and there is a cost of that certification associated to it. So that adds on to the cost. Then again, coming on to what Agilent can do is that we give you a value promise for 10 years. So which means that for the 10 years of your instrument use, you will be getting support from Agilent. You will be getting, even after the instrument model is obsoleted, you will get a value promise for an additional 10 years. So that and during that time, whatever spare parts or the support is required, Agilent helps with that. And apart from that, we also have financial solutions where you can rent an equipment, you can lease an equipment as well. So these are some of the other options that we have. There are many details to this financial services that we have. And if any one of you are interested, we can even talk in detail about it. So we have a separate team that takes care of this financial services. We also collaborate with government bodies like MOFP to help you with the fundings. So if somebody, if institute wants to buy those systems, then we have special academic discounts as well to help with the pricing. We also have this, our instruments are also built in such a way that they are modular in nature. So today, if your budget permits up to a certain level, we will customize the system and provide it to you so that it meets your requirement. In the future, when you have a more higher budget or you have more sanctioned amount, then you can add on the rest of the modules as per your requirement. And if we look into the cost of testing, so when I talk about the value promise, let us take the case of one of the major testings which happens in India, that is drinking water testing. So if I consider a lab which is doing 10 samples per day, so on an average, it will be doing for 22 working days a month, it will be doing 220 samples, and an average in a year 2,500 samples. So the cost of analysis which the lab incurs, I'm not talking about the cost of testing charge, I'm talking about the cost of analysis which the lab incurs in this case for testing 2,500 samples for a stretch of 10 years. If we compare the cost of a single quad GCMS, which is just enough to do the testing, so that would just be 30% of the overall cost of sample analysis. The major cost that comes in is from the reagents, chemicals, the lab personnel, electricity. So these are this is the other 70% which contributes to the cost of testing.
Yeah, thank you. My thing was because instrument developers keep developing, and then the lab wants something better and better. And I know Dr. Kaushik can vouch for that. Now coming to Mr. Ramgo, you are an entrepreneur, and we see a lot of the safe harvest goodies in our bags and thing. And from this morning, we have been hearing about the food safety culture. So in your organization, what is the role for testing of all? How do you view the current testing scenario in our country, and the competence of the laboratories who are doing the testing for you?
Thank you, Dr. Gowda. And thank you, Food Safety Works, for having me on this panel. I don't know whether I fit into this panel or no. And listening to everyone and, previous this thing, and fortunately, I'm not into foreign markets. So, for me, FSSAI matters, and I go by those norms. Well, at Safe Harvest, the understanding from the very beginning has been that unlike in the past when the organic food market relied on protocols and not on testing so much, we were of the opinion that for the consumer, what matters is whether the food is safe and whether it is evidence-backed. So testing is probably priority number one for us. But as everybody has been responding, that there is a cost to it. And in the early days, the number of laboratories who could do the kind of tests which were desired were very few. Over the years, the number of laboratories have gone up. There is also the aspect of, in the case of Safe Harvest, which works with farmer organizations at the back end, who are growing these crops without the use of synthetic pesticides, is about testing when the crop is aggregated. So these are difficult geographies. So to bring in a sense of credibility, the sample which has to be picked up from these aggregated commodities has to be by a third party. And the desired thing is that there should be a representative of the lab who should do this. And that has been a very, very difficult job and a very expensive job. So you have very small quantities sometimes, but to bring in this level of credibility into the business that I do, we incur a high cost because you need a third party to pick up a sample. I'm starting from the sampling itself. I'm not talking about the instrumentation part or even the testing part. And most labs find it difficult to do that, even at a cost. So that is one area that has always been a difficult area. And the second area is of the fact that, as you know, commodities which come out of a farm are not directly consumed. So they have to go through a process of milling and processing, etc., before finally being packed into retail packets and reaching the consumer. So there has to be testing at different levels. So one, you're testing when it comes out of the farm, then it's at the end of the value chain, and before it is packed. And this is an expensive sort of a proposition before you can this thing, because I don't own all the processing factories or mills that way. I go to a third party. So there are occasions when processors would agree, and there would be, because they would be doing jobs for others, including me. So we have to carry on with a certain kind of protocol before we can do that. So what I'm trying to emphasize here is to make it possible for a product to reach the consumer, retail consumer in metros, and to ensure that the product is contaminant and pesticide free is a very difficult and a very expensive job. So that is the kind of difficulty that we face, because at every stage almost, today we do batch-wise testing of a product. So we test it at the farmer level, then it's tested when it comes into storage after processing, and then finally before it is packed. So it is a 3-level process that we do to the same batch of commodity that which has been procured by my company. And I don't think there are many companies who follow such a stringent measure. And The unfortunate part is that because I don't carry the label, the certification of the fact that my stuff is organic or something, because I am relying on some other kind of a protocol, there is in the consumer's mind, because of the kind of marketing which is done of a certain product, etc., that's the fault not of the consumer, but I'll say of the marketing agencies, or the way produce is today sold in the market, a product. I carry the extra burden of having to explain to a consumer that my product probably has passed through a much stringent quality protocol than any other product which calls itself organic or is certified organic. So that's the area that I'm in. But I think we are kind of committed to the fact that we are okay about following the stringent this thing, as long as we are able to establish this as a category. And I'm happy to kind of inform and also take recognition of the fact that there are now many other enterprises who are coming into this category. So that's the happy part. The difficulties still remain, and I think as a family, or let's say of this ecosystem, the members of the ecosystem probably have to help us, kind of overcome some of these problems. Because, as ma'am said, and as you were also mentioning, that as these MRLs are becoming more and more stringent, as the instrumentation is becoming more and more expensive, and as the number of tests that have to be done to make sure that you call yourself pesticide free is becoming more and more expensive, because the MRLs are kind of being, becoming more and more stringent. So that's the area that is something that needs to be, kind of looked at or tackled or in some way explored much more.
Thank you. I think what I understand, from the testing point of view is the gray area is sampling. And that we all know, because as an analyst, having worked at Agent, we know that errors from sampling, if you haven't picked up a representative sample, 70% of the errors come from the sampling, and it's only 30% of the error which comes from the laboratory. So I think that is something in India we have to have either courses or thing for samplers and sampling, because not all laboratories have got people who have got experience in sampling. We don't have any course, as far as I know. ISTA does have some courses once in a way in India, but we do have some regulations. I think BIS has some sampling regulations which can be followed. Now coming to you, Vijay, you're from the industry as well as from testing, and ITC is one of the giants in processed food and thing. So what is your opinion on the way the testing is carried out by your laboratory? Because most of your testing, I presume, is in-house testing. And of course, you are NABL accredited. So what is it you foresee in the future in yours? And I know your sample load is very high because ITC produces so many products. So what is your opinion on, the testing scenario currently for industry? Yeah.
Thank you, madam. Thank you. First of all, I would like to thank Lalita Gowda, madam, and Food Safety Works. Thanks a lot. So, coming to your question, madam. So, first is the perspective what ITC is doing as of now for testing. So we do it in different stages. So the raw material stage, it will be, I would say, from farm to fork we test. So that's where ITC is, actually is the market leader in many segments, because we believe in delivering the quality products. So we never compromise on the quality of the products. So there is a program called ITC e-Chopal program, if most of you must have heard about it. So wherein we go to the farmers, we educate them on how to spray the pesticides and which level they have to spray. So from that level till we make a finished good and actually it reaches to the customer, we do take care in a lot of stages. So all the raw materials, we actually have now 450 plus products. So, and we have associated with more than 4,500 plus vendors for supply of raw materials. So from vendor approval process, so it is very stringent. So we actually demand for a lot of testing. So if the vendor is not a certified vendor. He will not become a certified vendor until and unless he produces a report from an enable accredited laboratory to us. So then actually once he becomes a vendor, so we have in 130 plus manufacturing locations across India. So wherein actually there is a quality control lab. That will be the first level of testing for all the raw materials as well as the finished goods. And there is also a quality assurance lab, so which is located in Bangalore. So wherein 80 plus scientists are working. And where we do a systematic sampling. So we go to all the factories, we pick up the samples. And also we tell them to send the samples periodically. And we are going to test that sample for all the residues. So that's how we are going to do, and it is a massive activity, definitely. So as you told, actually, so we are into 450 plus products. And going forward, actually, yeah, so what we are probably, what I foresee, so in the field of testing is, so the testing now is actually, the processes are very laborious, so and it is very expensive. So what we are looking at is probably, so now the world where it is moving, so the AI, so the new technologies, the rapid kits, so the rapid technologists, the kits which are coming in the market. So probably this will be the future solution. We may not have to, actually the amount of food what we are producing in India, so it is actually almost impossible to test. So if you don't get into rapid methods, rapid technologies, use of AI, blockchain, so these kind of things will come in the future. It is currently it may not be, so the real, it is not come into reality probably, but in few years definitely that will be the reality.
I have two other questions for you, Vijay. Now the trend is GCMS, MS, LCMS, MS, ICP-MS sort of thing. So when you have youngsters coming to the lab or thing, sometimes, we have to make them do mundane testing like proximate analysis or thing. And the younger generation also, wants to do something thing. So how are you coping with, when they come and they want to do only GCMS or LCMS and they don't want to do nitrogen estimation, fat estimation, proximate analysis or thing, because capacity building is required for the high-end equipment. Have you experienced such things where they don't want to do the mundane testing, which is very important, especially packaged foods, nutritional information we need to put onto the label. So in your testing. And the other question is, do you find the food technologists or food chemists or analytical chemists who come to your laboratory up to the mark to straight away start testing?
Yeah, to answer your first question, madam, yes, definitely we all face that challenge, because the generation what we are coming in, so they want everything instant. So they don't want to wait. So it is, but actually it is how we mentor them. So how we teach them. So we may have to actually, so make them understand. So what is the importance of routine testing? So when you get into the lab, so your hand should be set. Otherwise, we are talking about the residue analysis, we are talking about PPB, PPT levels nowadays. So the parts per billion, parts per trillion level. So there is high chances, if the analyst is not good, so there is high chances of giving false positive or false negative results, and which can be detrimental for any brand. So that's where actually we have to educate them, we have to, and actually there is nothing, analysis, if you take analysis, so there is nothing big, nothing small. So all analysis is analysis. So at the end of the day, it is adding to the quality of the product. So we need to make them understand that fact. And slowly, actually, so in the beginning, they may be reluctant to do that, but over a period of time, they will start enjoying. So, and when you show them the value of that analysis, what they are doing. And the second question is, so you asked about, so is the...
Yeah, the crop that you get in, do you feel they are ready, industry ready, or I wouldn't say industry ready, are they laboratory testing ready? Or you have to spend a lot of time training them.
Yeah.
Just say, like a fresh MSc analytical chemistry or a fresh MSc food chemistry, because a lot of them would come to you for jobs.
Sure.
So,
Yes,
yeah.
yes. So there is, we, as I told you, we have 300 plus scientists, and so we also have a lot of trainee pool. So output from the colleges, I would probably may not be they are ready for the industry. So there is at least 6 months of bridging need to be done. So that's where actually when we hire any fresher, MSc fresher, so we would take them as a trainee or actually as a FTC or intern. So we will train them. So we'll give them hands-on training. Wherein that period we'll give them stipend. It is not that we will not pay them. So we'll train them. So and we will make them tailor ready for our application. So whatever we want to do that. So we give the analysis, we will make them ready, and then probably we will absorb them. So I will not generalize that actually they are not ready. Probably few candidates are really bright. So such candidates we may hire, we will not wait till 6 months. Based on their performance, based on their eagerness to learn and their hunger. So it always depends on that. And few people may not, after 6 months also, they may not be ready. So we may have to extend their tenure.
Okay,
So it depends on the individual.
Thank you.
Madam. Thank you.
So coming back, Kaushik, I just have another question, because when we talk about MRLs, and because, I always say Dr. Kaushik is the father of pesticide chemistry or pesticide analysis in the country. He is the best in the country as far as I know in pesticide residue analysis. So, when we have these MRLs, most of the MRLs we have said for the primary produce, say for tomato, we saw the example giving. But how have countries, or have we, in India, have we come to extending those to processed? Because we cannot apply the same MRL that we have applied for tomato to a tomato sauce, or to a chili, to a masala or a garam masala or everything. So what is your opinion, and what are we at FSSAI doing about MRLs in processing?
This is an interesting question, ma'am, and this is also a gray area, not only in India but across the world. At this point of time, if tomato puri or chili powder or cottonseed oil, if anything is detected for pesticide, then the MRL applicable for the fresh agricultural commodity is directly applied. That is the general thing. So for example, green chili has MRL if it is 0.1 milligram per kilogram. Your dry chili is also will be having the same MRL. Same is the case for grape to raisin, or such many examples are there. But main problem is that there is a concept of processing factor which is still not in place in our regulation, but in FSSAI we are working on that. So what is happening now, in European Union, lot of work is going done. Now in European Commission, various laboratories, there are working groups who are extensively working on establishing processing factor. So processing factor means, for example, if 1 kilogram of grape containing pesticide residue of 0.1 ppm is dried to resin, then you first need to check what is the pesticide concentration in resin and pesticide concentration in the original raw agriculture commodity, that is fresh grape. So pesticide residue in the dried grape divided by pesticide residue in fresh grape, that is the processing factor. If processing factor is more than 1, that means there is an accumulation of pesticide residue. If the processing factor is less than 1, that means it has got degraded. So these are various concepts, and if it is equal to 1, that means there is no change in pesticide residue while processing. So same MRL will be applicable. So in this situation, unless we have proper information, we cannot apply for that. But in some cases, I can give example like grape to resin. In case of Europe, for example, if you have MRL for fresh grape is 0.01, they may use a factor 5.33 when they evaluate any pesticide residue in resin, because when grape is converted to resin, so almost 85% moisture is lost. So then they have came out with that factor 5.33, 15 divided by 85, 85 divided by 15. So that factor is used to multiply the concentration in the fresh grape to reflect what is the MRL for resin. So then that means if 0.01 is MRL for grape, for resin it will be 0.05. So that is the advantage. Because in case of processed commodity, we also consume a much less quantity. So if you take 100 gram grape, you are never going to take 100 gram resin. So that way, your daily consumption as well as risk associated with consumption and health hazard, those will be much less when you are actually taking processed commodity. So this is quite a complex subject, but FSSAI is working on it. And in Europe, a lot of data is currently available in various literature, even for cereals also, like rice to raw rice grain to rice, milled rice, rice flour, rice bran oil. So such sort of processing factors are available now. So this much only I can tell. But at this point of time, in our country, processing factor is not applied.
Yeah. Mr. Rambu, I just need, again, we talked about testing and you being an entrepreneur to do the testing. So when you choose a testing lab to do your testing, what is it you look for in that lab? Just like how what we are having is vendors. You have a vendor list and you're having approval and your thing. So I'm sure you also must be having laboratories which you send. So how do you approve of the laboratories for your testing to use it? I mean, maybe you don't do it, but someone in your organization does it. So.
But anyway, I think the topmost criteria is that it has to be accredited by FSSAI.
Okay.
Without that, we don't go to that lab. And the second, as I said, that it depends on the ability of that laboratory to give us services which go beyond just testing.
Okay.
And for us, sampling is extremely important. By the way, we work with farmer organizations. My organization has been set up by 8 farmer organizations. And these are in different locations. So they do need that kind of hand-holding in terms of they do not have the capacity. They're not a very organized, let's say, setup which can offer all kinds of services. They're not like regular suppliers of agriculture commodity. They do follow certain systems, etc. But at the end of it, somebody has to handhold in terms of how sampling has to be done. Those are issues which remain at that level. And they are also in very difficult geography, so it takes time to reach the laboratory, that sample. And you have to make it in a particular time, etc., etc. So I think that's the second part of the question that you asked me. That first is accreditation, and the second is the ability of the lab to go beyond just doing the test for us. So we do have a list of laboratories which have been working with us for quite some time. And third, definitely it's about negotiation on price, how much are they going to charge me. So that definitely plays a role. Yeah. And as the number of samples go up, laboratories are also willing to kind of offer these things.
No, the reason I asked you this question is because the misnomer has become that, everybody says NABL accredited. So many FBOs I know, they say we get it tested in an NABL accredited laboratory. But NABL accreditation is not for the laboratory. I mean, that's what I want to emphasize over here. It is not an accreditation for the whole laboratory like your ISO 22000 or thing, which is for the whole organization. The NABL accreditation is given test-wise. So that is what I was looking at, whether you will look at whether the test, say I'm doing pesticide residues, whether the laboratory has pesticide residues in their scope of testing. So many laboratories, I know many food business operators, I think, the thing is, NABL and ISO 17000 is like, go hand in hand, although it's ISO 17025 accreditation. So I just want to, tell the FBOs who get their samples tested or thing, it's not just an NABL accreditation or ISO 17000 you see on their board. You have to look into their scope to see whether for that particular test they have got the accreditation, it is in their scope of testing. So some have very limited scope. Some labs even with 10 tests get accredited. There are some labs with 3,200 tests things. So I mean,
that definitely
that
because,
scope is thing. And then maybe this is the last question I can ask Soma. In our country and among our analysts, or I think, the two words, just like accreditation, this verification and validation, there is so much confusion between, verification and validation. I just want to know, how does Agilent as a company try to educate the laboratories on, what is the difference between a verification and a validation for testing? It may be different in the food industry, but in testing we have these two words. So what is it you have done?
As a part of Agilent's programs for customer education, we do run regular webinars and workshops. So we send out regular invites where we focus on certain workflows or markets. So these are done based on different kinds of themes. So once it could be just made only on food, like in one of the sessions, ma'am, you agreed to grace the occasion. So we were talking mainly on food, about the new developments and how it is done. So those are regular programs that we run. There are educational resources already present on our hub. And most of our customer-facing people, for example, like we face most of our users in the lab, and then we have a field service organization who are facing our customers on site. So for us, we have a structured way. Whenever our customers are coming in to our lab, we go through an entire workflow process where we give them an overview about, first we try to understand what their scope of work is. So based on that, we tailor make the deliverables that we have for them, and then take them through a journey starting from the instrumentation basics to the entire workflow. So every time it is on a workflow approach. So Agilent stresses a lot on the workflow approach. And for most of our field service people, or those who are directly facing our customers at their site, so we have two dedicated teams. One team looks at only the hardware, which helps the customer understand their hardware on the systems, how to do regular troubleshootings and maintenance, which has got nothing to do with validation as such. But then we also have another team, post-sales support team, who will be going at your site and help you to run the applications. So they go through a full process of validation, and even when there is the discussion phase, when we are talking about the instrument and along with the instrument, what are the deliverables expected from our side, we have specific programs which are booked along with the instrument as a part of the order, where our people will go to your site and perform a method first run, which is only for setting up the application, where no validation services are provided. But then again, if our user wants, then we also include that as a part of package, where we have a complete hand holding over a period of 5 to 10 days, depending upon how many applications you want to perform.
Thank you. I think we just have time for 2 questions from the audience. If anyone has, yes, Dr. Sashikant.
Doctor, recently I think government enhanced the limits. I think, was it only for spices, or is it on board for all the products from 0.01 ppb to 0.1 ppb? In which case, so much equipment which is capable of measuring 0.001 ppb will go for a toss, because then a very ordinary equipment should be doing good enough to test, number one. Number two, as Agilent, when you have so much of good technology, can the testing bodies in this country who have the capability of high-end equipments do a CSR service for the country by voluntarily picking up samples from the respective markets and then, as Dr. Nimish said, come up with the risk assessment data, which can then plow back into the country's input. Third one is on the processing. So many vegetables or crops, especially, they undergo process of milling, baking, frying, and other things. So, what could be the restrictive level, and therefore, really going with the current MRL, would it be prudent enough to look at the risk of the processed foods?
Sir, what happened that when in 2018 December, the first that revised MRLs came, so out of some 210 or 215 MRLs, 129 MRLs were at limit of quantification level, 0.01. So India's regulation became so stringent that it was more stringent than European regulation in many cases. So situation became like that we can export, but we cannot sell it within our own country. But then FSSAI took a lot of effort that our panel rechecked all the values, because what happens that many of the pesticide companies, they had a feeling that pesticide residue should not be detected. So if something is not detected and that result is subjected to risk assessment, then what happens? MRL committee has nothing to do except fixing the MRL at limit of quantification. That was the biggest problem. So, but now the new MRL, which is going to be official very soon, that has most of the MRLs where, as per Suma Dasgupta, the lower models will be applicable. So India's economy will not be hurt in this situation. So many of the MRLs have been harmonized with Codex, harmonized with European Union, all the things. Second point is that regarding that risk assessment for domestic sector, this is already done by All India Network Project on Pesticide Residues for market samples as well as for various these things. And so that is a regular activity which is going on. And one report you will find in FSSAI website also, 2019 annual report. So that is a nice report which compiled almost 24,000 sample test report. Similarly, milk survey was also done by Bimta Laboratory, was the lab who tested all the samples. More than 4,500 samples were checked. So such type of things are happening in our country. And what was the third one?
I think very recently 2,000 spice samples have been analyzed after the ETO, and before that 24 samples of MDH and things were analyzed for ETO, and ETO was not found. I think when the report came in 19 samples, because laboratories have to submit it, it is there. This surveillance as such, what you're talking about, the data, FSSAI does it. You can find a lot of reports, even aflatoxin in milk, fats and oils, so that is done. But then there we do not take the help of corporate laboratory, laboratories that, because we think, it is, there will be a lot of RTI. So we always take only the FSSAI notified laboratories, the referral laboratories, to do the analysis. Any other question?
But one question, sir, you asked that increase in MRL for ETO. Actually, this is not MRL.
Yeah.
What happened that in our country, ethylene oxide has to be tested by most of the labs to take care of the current demand. So under this situation, it was decided that 0.1 could be limit of quantification.
It's only for ETO.
Only for ETO.
No, for spices, it has not been changed. Actually, what you asked was for spices being, I think, the media reported that, MRLs have been increased.
Absolutely.
But actually, it was the original MRL, which is the same as Codex. So it was harmonized with Codex. But that is what happens with media, they hype everything. And that's what happened with ETO also.
See, another thing is that most of the SME depend on mandi purchases, well aggregated.
Yeah.
Now, there are very good examples in ports where there are testing laboratories. In a similar way, can the country look at, at APMC level, setting up labs so that a buyer can use the lab facility even for little payment, ensure that he buys the right quality, from the mandis. That's where nearly 70% of the purchase happens.
I think it all amounts to again coming to either a private laboratory or a government laboratory or a state laboratory, whichever is closest to you. But I think the answer to that is the rapid tests, what we were talking about, as if we can have a large number of FSSAI approved rapid kits where they can just, put the strip, see whether pesticide residue is absent or not, screening. I think that is what is really required. But again, we need it to be approved. Not everybody makes a kit and sells it in the market. Just like when COVID was there, we had so many kits and we didn't know which to believe. With this, I would like to close the session. Okay, please go ahead.
Yeah. Good evening, everyone. So, right now we are discussing about this pesticide residue and heavy metals. So, what are we are doing? It's everything is the lab-based technology. So, what about fruits and vegetables? So, each pesticide having some tolerance days. After that, only farmer need to harvest.
Yeah.
So, nowadays, due to the pest infestation, farmers doing the pesticide application and immediately harvesting the material, bring it to the market and the retailers. So what is the method? Is any method developed to check, instant check, like litmus paper or anything?
No, that is up to the farmer to do it. You see, the MRLs are based on, the last spray after 15 days. Only after 15 days they should harvest. That is good agricultural practices. But I don't think the government can have a hold on telling him that you cannot spray 2 days before bringing it to the market. See, the only thing, and if you look at FSSAI regulations, the farmer and the fisherman are exempt. So whatever you test at the farmer thing or with the fishermen, you're not allowed to, they don't come under the regulations. So once that comes into the hand of a third person, then that person is liable. So it is the responsibility of that person to ensure that the product, does not contain the pesticide. You have to remember our act, our Food Safety and Standards Act. The farmer and the fisherman are exempt from the act. So you cannot test any of their produce and prosecute them at their farm or the fishermen. Yeah. Okay. Right? I hope, I think you didn't know that. The farmer and the fishermen are exempt.
Right, but issue start from there only.
Yeah, issue starts from there, but our act says that.
That purpose, we need to put some restriction, like while receiving itself. So basic test, if they are initiating, means farmer also get know our product is tested, so we need to reduce the pesticide application before harvesting.
Yeah, that is what responsible sourcing is, like Mr. Agrawal is doing. Like, you source from farmers who are following good agricultural practices, and, we don't have the pesticide. Most of the FBOs, they do this type of sourcing. They source from responsible farmers.
My request is, if able to do that, please help the developer and researcher.
I don't think I can modify the act.
It is only 10%.
Yeah.
So organized retailer doing this kind of putting agronomist and all, it is only 10 to 20%. Remaining 80% market material is coming like that only.
Sure. I'm sorry. Act is like that. I think Sarika had a comment. Okay, farmer and fishermen are exempt. I think there is some talk about, including the thing, but it takes a very long to change anything in the act. Yeah, act and rules. Yeah.
Something that's spoiled somewhere.
Yeah.
Nobody can fix it.
Yeah. Okay. So if this is not there, I would like to thank Dr. Vijayan and Dr. Rangurao, Kaushik and Soma for, this discussion. And I'm sure now all of you know the testing, what is there, how difficult it is, and, problems. And of course, the importance of having your raw material and ingredients tested to meet our rules and regulations. Thank you, everyone.
I'd like to request Dr. Lalita R. Gowda to honor all our panelist members. Small token of appreciation to Dr. Soma Gupta. Dr. Kaushik Banerjee, Mr. Rangu Rao, and finally, Mr. Vijayananda V. And I'd like to invite Mr. Srinivas KR to present a small memento to our moderator, Dr. Lalita R. Gowda. Yes, sir, please. A round of applause for all the panelist members. We hope the session was very insightful. Thank you. Thank you. Thank you. Thank you. Thank you. We will now break for a coffee and we will reconvene in 15 minutes. Please reconvene by 4:15, 4:20.